Okay, we're here. Hello, welcome everybody. Welcome to the stream. I have been dying to get to this paper and also dying to stream because I didn't get to stream last week, so I missed you all very, very much. Good morning, Kevin. Hello, Ramsey. Welcome, welcome. Hello, the Mighty Monkey. Welcome. Hello, Alexandros and Ari and Flying Taco. Hey, Clockwork. Welcome to the stream. Welcome, welcome. Hello, Seed Yum. Anyone else I'm missing? Hello, Rain. Welcome, welcome. Yeah. A very special paper that I have been dying to talk about, being in my primary specialty here, given that it is both concerned with the immune system and the nervous system. And I'm very, very excited to kind of relive what it was like learning about microglia for the first time. And just as the progress of science marches forward, we are learning more and more about what these immune cells do. And I hesitate to even call them immune cells because more and more we're realizing that these specialized macrophages they can just do everything. I think somebody in the Discord had mentioned that, you know, they would no longer be surprised in terms of what we learned that microglia can do. So I'm really, really excited to show you kind of how amazing these little cells are. Okay. So let's go ahead and just jump right into it. Oh, and welcome, Ludi. Welcome, welcome. Okay, so let's get started. Okay. Alzheimer's has a host, sorry, it's early in the morning. Alzheimer's has a whole host of potentially overlapping mechanistic origins, right? So, there's been a lot of debate and even controversy with regards to what causes Alzheimer's. And so in terms of having the most generous view of what could be causing Alzheimer's, we can just list a whole bunch of different modalities that people are thinking about in this space, including things that have to do with how damaged cells are, so dendritic damage, things to do with the immune system. There's a pretty interesting theory that Alzheimer's is like a type 3 diabetes where your cells in the brain start losing sensitivity to insulin, and that can cause a whole host of problems. Metabolic origins, cholinergic origins, so having to do with specific neurotransmitters in the brain. But a lot of attention has been given to a subset of these hypotheses. And again, these mechanisms don't necessarily, they're not like isolated bubbles, right? They can be mutually inclusive. So they're not mutually exclusive is what I mean to say. But in terms of like where a lot of attention has gone towards, it has been towards these aggregating proteins. So many of you might be familiar with an aggregating protein called amyloid beta. So this has been the dominant hypothesis for Alzheimer's. Those of you who are in the know that that hypothesis has been marred with unfortunate scientific controversy. That doesn't mean that it's not true. It just means that it's difficult to make heads or tails of that data. All right. Yeah, microglia are immune cells. They are cells of the brain, but they are also a special type of macrophage, meaning that they have a shoe in both worlds, and that's why I love them so much. All right. So let's go over just what the star of the show of today is, and that is this protein called alpha-synuclein. So like amyloid beta, like tau, this is a particular neuronal protein that is aggregated in neurodegenerative diseases such as Parkinson's and Alzheimer's, right? So this is a particular synaptic protein that is potentially involved with vesicle recycling. It is intrinsically disordered, meaning there's a lot of floppy spaghetti bits here. So this protein does not have a neat organized shape, meaning that it is very prone to aggregation. If you've watched my prion video, and I can't imagine if you're watching me stream that you haven't watched that prion video, you would know that these disordered proteins are really, really bad for the nervous system, essentially. And so these synaptic proteins aggregate in little subcellular compartments called Lewy bodies that can cause neural death, right? So here we see an example of a stained Lewy body, a aggregation of these floppy, intrinsically disordered proteins. So these are no good, right? So I kind of clickbaited you all with the title because I had mentioned that this was going to be an Alzheimer's paper, and in ways it is, right? So alpha-synuclein is hypothesized to be a part of the Alzheimer's story, but this paper in general is taking a much more narrow view on Alzheimer's. So in terms of direct applications to the disease, we won't really know for certain in the scope of this paper, but what I can say is that this is an Alzheimer's protein paper, right? So just wanting to get that clear, right? The scope extremely clear. Okay. So the other main character of this story are these cells known as microglia. So this is the brain resident macrophage. So this is in terms of the actual lineage, it's not entirely clear, but they are essentially the macrophage of the brain. And so these are supporting cells of the brain, the main immune defender of the brain. So if you have brain trauma or an infection in the brain, microglia are going to be there to fight off that infection or try to repair that damage. So they have key functions in healing during brain injury, synaptic pruning, and housekeeping. I remember when I was first learning about microglia in stem cell school and, not really thinking too much about them, that, oh, they're just janitors of the brain. They clean up debris and dead cells or whatever. And then at some point in my grad student career, I had learned that microglia actually have a nervous function, like a nervous system function. They eat extra connections between neurons in the brain, and I found that to be incredibly interesting, right? And I think this has been the theme of a lot of my training, is that I really love stories that are unexpected. And so it's really unexpected that this immune system cell that has been specialized to the nervous system is affecting your cognition, affecting the way you think and feel and memorize, etc., by eating away at the connections that your neurons don't need, right? So when you're born, you have an excess of synaptic connections. Your neurons have way too many connections to each other. And over time, those connections get pruned in order to make your brain more efficient at what it needs to do. And so instead of it being reliant on the nervous system, this function is provided by an immune system cell. And I love that. And so given that they do eat synapses and debris, this is a professional phagocyte, meaning its main function is, or one of its main functions is to eat up stuff. And I find that really, really cool. So the protein aggregate versus microglia matchup is at the heart of many neurodegenerative diseases, right? So, we humans, whether we like it or not, live to the point where we can develop neurodegenerative diseases. Oftentimes animals don't have lifespans where their brains start to get diseased. This has been a very important issue in the field of neurodegenerative disease because mice, the most common, most popular, most useful animal model, they don't get Alzheimer's. And part of why that might be is that they just don't live long enough for protein to ever accumulate. And so to understand how our brain clears these protein aggregates, whether it be tau or alpha-synuclein or amyloid beta, is going to be very important to understanding how these diseases actually work. All right. However, for a long time, it was thought that microglia could only clean up extracellular junk, meaning that the microglia would always be playing catch up with dying neurons. Remember, neurons are accumulating these protein aggregates over time, and we have good reason to believe that that causes their death, right? So you start getting these tangles of tau, you start getting these fibrils of amyloid beta and alpha-synuclein, and neurons start dying. And if all microglia do are clean up the aggregated protein after the neuron's dead, then they're always going to be playing catch up, right? And so while that's useful, right, you don't really want excess garbage in the extracellular space. It meant that I think for a long time we weren't really sure whether or not microglia were capable of alleviating the burden of these aggregating proteins. Breed older mice, larger brained mice, harder than you would expect, right? So if we were capable of breeding older mice, then a lot of questions about longevity would already be answered, right? So the average lab mouse lives at most 2 years, and that just isn't enough time for anything to... That's why when you do study Alzheimer's in mice, you have to genetically modify them to accelerate their neurodegenerative disease, and if you do that, there are artificial elements that might not be captured in those disease models. Are animals that live longer than humans theoretically able to get Alzheimer's? That's a really interesting question. I don't actually know whether or not, for example, gorillas or chimpanzees can get Alzheimer's. It'd be worth taking a look into for sure. So, again, it was thought that microglia were just there cleaning up the corpses of dead neurons, cleaning up the junk in the extracellular space, but then this paper in 2021 had found that microglia actually work together to destroy alpha-synuclein cargo by distribution through something called tunneling nanotubes. So if you do exclamation point jargon, you can kind of get a quick definition of that. But I want to highlight this kind of game-changing finding, right? Again, to set up the stage, microglia, as far as we knew up until 2021, only dealt with things outside of cells. And this paper shows that there's a possible connection that microglia can make towards each other where they can transfer proteins between themselves through these tunneling nanotubes. So here on the left, we see a microglia that is suffering. It's eaten up a lot of alpha-synuclein and it's dying, right? It's causing oxidative stress. This microglia is signaling distress with its inflammatory response. It's going to die. And what was found in this paper is actually that neighboring microglia can help alleviate this burden, suck up some of that alpha-synuclein, and also donate healthy mitochondria that haven't been damaged through these tunneling nanotubes. And that is absolutely crazy. That is bonkers, right? Like the fact that this is now a game-changing finding, that there's this connection, and it's not done in a way that is more typical for cells, right? Cell-cell contacts generally have a flavor or a flow between them, and this tunneling nanotube thing is something that I've personally never seen before. So very, very interesting stuff here. So here's some data from that paper, and you can actually see these are two microglia, and in red we have labeled alpha-synuclein. You can actually see that this microglia on the left here is sucking up alpha-synuclein from this particularly burdened microglia on the right. Now, I don't actually know why this microglia that, I mean, it looks like it just has more alpha-synuclein, why this microglia is sucking up from this smaller microglia, but I imagine this is maybe a more immature microglia that has a relatively larger burden of alpha-synuclein. But the point is that over time, when you take a video of these microglia, you can actually see a very gradual transfer of that alpha-synuclein to the different microglia. So this is, again, game-changing. We thought that they could only take up these corpses from the extracellular space, but they're actually capable of passing this aggregated protein between each other. And that's pretty nuts. Thank you for subscribing, Kendra. Welcome, welcome. All right. It's the sharing of mitochondria that gets me. It's such a smart way to combat oxidative stress. Yeah, and we're going to get into the mechanism of that in a bit. But I just want to highlight another incredible picture. It kind of looks like we're looking at galaxies here. A much more well-resolved picture of two microglia that are sharing mitochondria as well. So here we have a fluorescent image, phalloidin. This is an actin stain. So this is a toxin that actually binds to actin, actin being a component of the cytoskeleton and a known component of these tunneling nanotubes. So these tubes that these microglia use to communicate with one another, if you stain that phalloidin, you can actually see that connection a lot better in that other picture. And then what we can actually see in red here is alpha-synuclein, and in green, we have mitochondria. So within this tube, if we blow this up and crop it, you know, it's really hard to see in the overlay, but if you isolate the individual fluorescent channels, you can see alpha-synuclein in that tube and mitochondria. Just absolutely crazy. Question, can they extract alpha-synuclein from other types of cells? Excellent question, Alexandros. That is going to be the topic of today's paper. Remember, this is just the background. This is just the background. This is the opening act for what we're about to see. So if microglia can share cellular... here's the slide. If microglia can share cellular contents through a nanotube, are microglia helping neurons in the same way? So tunneling nanotubes, they're not new, because I was looking into the history of them, but they're new to me, and they're new in the context of microglia, I believe. So this new connection that forms between microglia, can they use this tube to actually help neurons directly instead of just passing off the burden between each other? These nanotubes remind me a lot of these pili that bacteria have. I just wanted to point that out, that when I looked at the structure here, this tiny thin tube. Again, this is a pretty unusual way for cells to be sharing things. Usually it's through gap junctions where they are more, where they have more surface area touching each other. And so this strategy reminds me a lot of how bacteria share genetic information, how they share DNA by forming these very thin connections where they can extrude and receive DNA. And I just think that's kind of neat. Convergent evolution, one may think. Okay, so that brings us to today's paper. Microglia rescue neurons from aggregate-induced neuronal dysfunction and death through tunneling nanotubes. So Alexandros, that's a bit of a spoiler for your question here. The answer is yes, and we are going to be taking a look at how they do that. Okay, first let's kind of go over how alpha-synuclein hurts neurons to begin with. And if we take a look at the genes that are enriched in neurons that are treated with alpha-synuclein, we can see that there's an enrichment of oxidative phosphorylation, as well as genes that are associated with various neurodegenerative diseases, right? And so what we can take away from this is that alpha-synuclein, as it starts aggregating in neurons, is going to stress out the mitochondria, right? Because the mitochondria are the organelles that are helping your cells prevent those dangerous oxygen molecules from damaging other parts of the cell. So to rewind, right, just to give context for this, oxygen is an incredibly dangerous molecule. It is so bad for anything organic and is responsible for one of the greatest extinction events in all of biological history, essentially, right? As soon as those, one of the cyanobacteria started to extrude oxygen into the atmosphere, it caused the most catastrophic loss of life in our planet's history, I think, probably. Because oxygen is a very, very reactive molecule. It's why things light on fire, right? Like you can imagine little bursts of chemical activity when molecules are exposed to certain forms of oxygen. And so the mitochondria have evolved to use that oxygen in an appropriate way and then to convert it into something much safer, i.e. water, right? So if you have oxygen that isn't being able to convert into water, you can potentially have a leak of dangerous oxygen species that can hurt cells. So And if you ask the question, do we actually see stressed neurons when they have alpha-synuclein added to them? The answer is yes, right? So here we have the mean fluorescence of reactive oxygen species. So you can actually label the harmful forms of oxygen with a fluorescent marker, and you can actually see that neurons, when they are given alpha-synuclein, they get really stressed out compared to the control, right? So these are just two different ways to measure oxidative stress here. TNT, that stands for tunneling nanotubes. I'll go into a little bit more about what that is in the future. But essentially, it's a very, very thin contact that microglia share with one another. Smaller TNTs don't even have microtubules. Yeah, I know. They're really weird, and I really want to get into what makes them tick. It's really, really interesting. All right. So yeah, here. So leakage of the mitochondria leads to oxidative damage. So the mitochondria has two layers, and in this inner layer, we have a lot of the protein machinery that Clockwork is very, very familiar with that help pass dangerous species of oxygen to various enzymes to convert them into water, right? So when it's water, it's not really a big deal. But if that O2 minus collides with some other organic material, that can really, really damage whatever it comes into contact with. So it relies on these enzymes to first convert it to hydrogen peroxide, which then gets converted to H2O. But if this conversion between O2 minus and H2O is not complete, if it stops at O2 minus or H2O2, these molecules can then leak out of the outer membrane and cause serious damage to the cell, right? So alpha-synuclein is doing something to the mitochondria that is hurting them and causing stress and damage in these neurons. So that's sort of the one mechanism by which alpha-synuclein can damage neurons. So TMRM is a mitochondrial membrane integrity marker, right? We were just saying how mitochondria that are damaged are leaking harmful oxygen. And I just want to ask chat if this data is consistent with the story so far, right? So on the left here, we have a control neuron, and the whiter the pixel intensity is, the more that it is biased towards the yellows and the whites, the more intact those mitochondria are. So I just want to ask chat, is this data consistent with the model that we have so far? When you label the control neurons and the neurons that have been treated with Alzheimer's protein with TMRM, do we in fact see that the neurons that have alpha-synuclein have damaged mitochondria? What does that look like to you, chat? Pretty good opportunity to get used to interpreting some fluorescent data. What do you think? What do you think? Do we have damaged mitochondria in the alpha-synuclein treated neurons? Do we see that kind of damage, do you think? And if you're unsure, feel free to ask questions about what it is we're looking at, because there are a lot of neat fluorescent images that I would like to share. And it's just kind of more fun if we're all making attempts at interpreting the data for ourselves. Clockwork says the left one has more intact mitochondrial membranes, right one has less intact, right one is labeled alpha-synuclein. Yeah. That is what I would interpret as well. Yeah. It looks like it, but I wonder why the integrity isn't uniform throughout. That's because mitochondria are not necessarily localized in the exact same location throughout. I don't really know what causes asymmetry of mitochondrial distribution in neurons. It's a pretty interesting question, whether or not the intrinsic shape of the neuron demands more energy production be in certain locations. Certainly interesting idea behind that. So it looks consistent to me. Yeah, it does to me as well. Welcome, Robert. Welcome to the stream. What does it mean to be more intact? It's a great question, Super Soyajin. So if we take a look at the mitochondria, so this is a zoom in, a cross-section of the mitochondria. The mitochondria has two membrane layers, one that contains all the machinery that convert harmful species of oxygen to water, and in that process, help you make ATP or energy. And then we have an outer membrane. And so if you have damage to the mitochondrial membrane, this outer membrane, sorry, my cursor's not on screen, this outer membrane can be compromised and leak harmful oxygen into the cellular environment. And so what we're seeing here with this membrane label is that In the control neurons, you see a lot more of this stain. So this is marking a mitochondria that is intact, that has not had this outer membrane damaged in any kind of way. But when you introduce proteins that can hurt neurons like alpha-synuclein, you see a lot less of that stain taking hold, a lot less of that dye, meaning that these mitochondria have had their mitochondrial membranes at least partially disrupted. Great question. Yeah. So this is adding up to a story where in neurons that are burdened with this Alzheimer's protein, their mitochondria are hurt, which is going to set up a really beautiful experiment coming into the future. So here are the results so far. Alpha-synuclein, again, this Alzheimer's protein, causes oxidative stress in neurons. It's creating oxygen species, it's stressing out mitochondria, it's damaging mitochondria so they can no longer turn that harmful oxygen into water and prevent your cell from being damaged. So then the next question is, given what we know about how microglia help each other, can they help neurons in the same way? So we already saw in that background paper that microglia can actually share mitochondria, that a very stressed microglia can receive healthy microglia from a neighbor, and that's pretty incredible. Harmful oxygen is also the exact same mechanism that causes nuclear radiation, uses to kill you. Ionizing radiation shatters water molecules, creating reactive oxygen species and those wreck cell components. Exactly. That's why, all these health people want you to eat blueberries, right? Any foods with high levels of antioxidants can help with reducing the oxygen burden that your cells have. Okay, so the question, can they help neurons using tunneling nanotubes? Can they share in their burden? And here we have a really amazing EM image. So this is a technique called electron microscopy. The wavelength of visible light is actually too big to fit in between some of the cellular processes. So instead of using visible light, we are using electrons to image these images. And so by using electrons, you can get a lot higher resolution here, which is why, when you look at fluorescent data, it's always so blurry. But when you use electrons, they're really, really tight here. Do all mitochondria eventually have their membranes damaged with time? That's an interesting question. I'm not like a mitochondrial expert, so I actually don't know what happens to damaged mitochondria, whether or not they have their own like apoptosis going on, or whether or not the host destroys them in a certain way. Be very interesting to look into that more deeply. I imagine because they are dealing with literal molecules that damage organic material, that they are just reactors that are waiting to burst. That would be my hypothesis, at least. So, what are we looking at here? This little hairy guy is a microglia and is surrounded in a mess of neurites. So neurites are these little membranes or little branches of neurons, right? So this microglia, if we zoom into this area, we can actually see the microglia form these tunneling nanotubes with surrounding neurites. And if it's surprising that these neurites are all over the place, it's because neurons can be very, very long, right? So this is a neuron, and this neuron is making tons of different little neurite protrusions all over in this network. It's impossible to know which neurites belong to which neurons, but what is known is that these tunneling nanotubes are attaching to these neurites, right? So that's really, really exciting, because there then becomes the possibility that the microglia are donating mitochondria to the neurons as well if they're forming these tunneling nanotubes. But of course, scientists have to be very thorough, right? We can't assume that these microglia are in fact donating mitochondria, but this is a really, really interesting start. And also just look at how beautiful this image is. It is so crisp and amazing. It's just incredible work done by these people. Okay. But while EM is really good at imaging like with your eye, it can't actually get you stuff inside the cell, right? And so in order to do that, we need to go back to fluorescence, which is a lot less higher resolution, but can tell us a lot more information about how things are working. And as I was reading about tunneling nanotubes, they're apparently very delicate to image, right? Like you can barely see them the connection between microglia and neurites. So let me explain a little bit about what we're looking at. So WGA, this is a cell marker. I believe it's a marker for microglia. F-actin, this is a component of the cellular cytoskeleton, so the bones of the cell, if you will. And then cell tracer, I believe this one is a cytoplasmic stain that stains the inside of the cell. So you can see here that if you zoom really close, you can see this very thin connection that we've seen before in those background papers. And I want to take a moment to really appreciate the work that was put into this paper. And I want to make this clear. Last week, or 2 weeks ago, we had covered a kind of not so good paper about aspartame published in the journal Cell. And I want to make clear that Cell publishes incredible work most of the time. And I really want to drive home that aspartame paper is not the rule, it is the exception. Here we have something that I think is much more characteristic of Cell, because I've just had my jaw on the floor the entire time reading this paper. And the other reason why that is because I've actually done experiments similar to what they've done here. And the thing about imaging very fine ultra-structural components of the cell is that these cells have to be super healthy in order to have these microprocesses, right? So to give you a bit of context, they took fetal pups, mice, and took out their brains and isolated a particular subset of those neurons, and then took those neurons and plated them on a dish, and that is what we are seeing here. So the reason why this is so hard is because neurons are what is known as post-mitotic. They don't really do mitosis, right? So if you isolate the brain cells, you don't get more. You get what you get. And if you damage them or hurt them or stress them out along the process of isolating those neurons, they're just not going to be as healthy. And when I was working with neurons, I just found it took me like 2-3 years to be able to get not even good quality neurons, but like acceptable quality neurons that made some of these micro structures, right? And so the fact that they are able to get these tunneling nanotubes to even form to begin with means that these microglia and these neurons are very, very happy and healthy. Absolutely insane. Okay. So just wanted to shout them out. The people that were doing this work probably had like really, really dexterous hands to get this to work properly. Okay, so taking a look at sort of some basic data as to analyzing the relationship between these tunneling nanotubes and these neurons. This is just like exploratory data to see what's up. And what they had found was that microglia make more tunneling nanotubes with neurons that were treated with alpha-synuclein, right? So when you add microglia to regular neurons, they make a certain baseline level of tunneling nanotubes. But then when they sense the neurons that are burdened with alpha-synuclein, they react by making more of these connections. And that's really, really interesting to me. So there's some component of feedback here where the microglia understand molecularly that there are stressed neurons nearby. The lengths of those nanotubes seem to be the same, though. I think this is just interesting to know that like this is maybe a set kind of structure, that this tunneling nanotube maybe only comes in one flavor. And interestingly, this doesn't happen with tau. Also, there's a typo here with the length. That's kind of interesting. Gotta head out. Have a good stream. Thanks for dropping by, Super Suasion. Yeah, so this effect with the tunneling nanotubes seems to be specific for alpha-synuclein and not to tau. So they don't seem to be reacting to the presence of tau, but only alpha-synuclein. And that's a head scratcher for me. What's the percentage with 4 sigma again? I have forgotten. I'm not entirely sure what you mean by that, Alexandros. You mean the 4 stars here? This basically means that the difference between these two groups is statistically significant. And then if it doesn't have those stars on top, then it's essentially statistically the same. Do microglias only collect and store alpha-synucleins, or do they dispose of them? You are jumping ahead of me, Ramsey. Stick with me, and we will find that out together. So, microglia make more tunneling nanotubes, connections with neurons that were treated with alpha-synuclein. So here are the results so far. Alpha-synuclein treatment causes oxidative stress in neurons, potentially due to damaging their mitochondria. Microglia make more tunneling nanotubes with neurons that have been treated with alpha-synuclein, so they are reactive in a way. And then do these nanotubes help unload alpha-synuclein from neurons, right? So we know that they attach, we know that they react, but are microglia actually sucking up the alpha-synuclein? That's something that we have to prove. So here is some fluorescence data. Here we have a microglia and a neurite here. And if we zoom in here, what do we see? Again, the overlay is really hard to see, but in green we have a microglia marker. In red we have a marker for the actin cytoskeleton and the tunneling nanotube. In white we have cytoplasm being highlighted. In blue we have alpha-synuclein, and then inverted alpha-synuclein we'll see in just a bit. So while this overlay isn't exactly helpful, even though that there are arrows here because all these colors are just mashing up on one another, if we just take the alpha-synuclein channel and invert it, we can actually see that yes, we do have in these tunneling nanotubes a bit of alpha-synuclein making it into those tubes. Very, very interesting. Is there a known mechanism as to how microglia know that there's extra alpha-synuclein in the neurons? As far as I know, there isn't. As far as I know, because this paper was released in 2024, so I'd be really surprised if someone already figured that out. It could be something that some of you find out one day, you students in the audience. Yeah. I would love to know how they detect alpha-synuclein burden. It goes to show that, like, when I was in high school, I was like, how much more biology is even left? It feels like we've solved everything. And at this level, it's like, no, we're always finding out more. And that's what's so exciting about science. If you look at the tau, I don't really think it's as prominent, but they're making the argument that tau is also in these tunneling nanotubes because it's here right at the beginning, but I'm not super convinced because I view this as the tunneling nanotube and this potentially is just another part of the microglia, but you know, I'm going to shrug my shoulders. Tau isn't going to be like a major component of this story to begin with. Cultivating neurons looks painful. Yeah, it was really, really hard and you have to be very good with your hands. I would not recommend it if you aren't very careful. Okay, so those were still immunofluorescent data, but this is just a snapshot, right? This is a moment in time. And here I have a very, very, very exciting video here. A very, very exciting video, even though there wasn't any significance with the tau. So they appear to be deaf to the burden of tau. That doesn't necessarily mean that they're not also helping with tau. It's just that they can't detect if there's tau around. It's a little nuanced and a little confusing, but that's the best way that I can put it. But yeah, so here is a video, and in green we have alpha-synuclein. Down here is a neuron, right? Down here is a neuron, and then these up here are microglia. And we have an actual video. We have a video of the microglia sucking up the alpha-synuclein. So let's go ahead and just watch this a couple times together. So here, this is the channel that's a little more informative. You can actually see that particle of alpha-synuclein get taken up, and this microglia kind of drag out that alpha-synuclein. Absolutely incredible. Look at that. The rest of this video is kind of samey, so let me just replay that. So again, down here in green, we have labeled alpha-synuclein. This is a neuron, and then the more spherical guys are the microglia. And we want to pay attention up here to this dot that gets sucked right up into that microglia. And then down here, there's another dot, and then this microglia kind of rips that alpha-synuclein right off of that neuron. Very, very, very cool. Just amazing, amazing video. I love that so much. And we'll just watch it one more time without me interrupting. It's just... damn. And the resolution, right? The quality of data that you need to be able to resolve at this level is just breathtaking. Okay, so what do we conclude? These tunneling nanotubes contain alpha-synuclein, the culprit for certain neurodegenerative diseases, and it does look like when you take a video that microglia are capable of tearing out that alpha-synuclein. However, are we convinced right now at this point, chat, that microglia are doing this in any meaningful way? What are some alternate hypotheses here, chat? I want us to think critically and challenge the paper and not buy too much into what the authors are selling us. Like, even though this paper has been amazing so far, I want to cultivate a mentality of skepticism. So we just watched that video of microglia tearing out alpha-synuclein out of the neuron. Do you think that this is sufficient evidence to say that microglia are helping these neurons with their alpha-synuclein burden? It's like a tiny straw. Exactly. It's exactly like a tiny straw. So crazy. I know. It's incredible. But yeah, and I don't want us to fall too much in love with what they found, right? Because that's the danger of reading a paper that you love so much, is that you begin to lose your sense of skepticism. So I want us to remain grounded. Even though what we witnessed was amazing, are you convinced at this point that the microglia are in fact reducing the alpha-synuclein burden in neurons? They could be targeting something else. Yeah, hair day. So maybe they're sucking up something else, and maybe the alpha-synuclein is just there for the ride, right? The process may have a downside that leads to further long-term damage. That's also true, right? This has been shown to work in a dish, right? That this is theoretically possible, but whether or not it's meaningful is a separate experiment, right? So I want us to keep that in mind, that in vitro, meaning doing things in a dish, has a particular advantage because you can control everything in a dish, but you can't control it in an animal, and you can't control it over the long term, right? So that doesn't mean that in vitro experiments, and you'll see this all the time on Reddit, because Reddit, whatever, that in vitro experiments tend to get a lot of flack from Reddit. Oh, you cured cancer in a dish. Like, whatever, just add bleach. That kills cancer too. Like, whatever. There's not, and I think that's such a cynical outlook on biology research. And it's not their fault, these Redditors. If they haven't had a Phi the Neutrophil stream, they might never appreciate the fact that everything starts in a dish, and it's still worth celebrating. It's just that you have to be ready to get hurt every single time, right? So I just want to course correct on that kind of attitude. With these rat cells, I think they're mice. How would an in vivo experiment go? Well, we'll look at some in vivo experiments in the future of this paper. But yeah, so while what we witnessed was pretty amazing, we have to keep our skeptic hat on and go further down the line, right? So do microglia actually remove alpha-synuclein from neurons, or were those images just, luck, essentially? So neurons alone are not good at degrading their alpha-synuclein, right? So if you add alpha-synuclein to a neuron, you actually see that when they take up that alpha-synuclein, that they're not able to deal with it. That's what you get in this chart here. So uptake, this is when you give the neurons the alpha-synuclein, you wait for a period of time to check for degradation, and they just don't degrade the alpha-synuclein. Neurons are actually really bad at that. However, when you add microglia to the neurons, what do you see? You see that when you add the microglia, that you get a reduction in the alpha-synuclein fluorescence, about 50% of the original. That's pretty interesting, right? That's pretty interesting. You give the neurons the microglia, and it reduces the fluorescence by about half, meaning that there is evidence to show that microglia are actually helping, that this is meaningful, that not only can this happen, but it does happen, and it happens enough where it could be meaningful, right? Very, very exciting stuff. And they did an extra experiment that I think is really, really worth doing. And that's to add an insert in between the microglia and the neuron. I didn't really have the time to illustrate this, chat, so I'll try to paint a picture for you. In this red graph, right, what happened was you took a bunch of neurons that are in a dish, and then you dumped microglia on top. So the microglia are able to have a direct contact with the neuron. And what makes this experiment so beautiful is that what happens in this, I don't even know what to call this, pinkish bar, what happens here is you have a dish of neurons that you put directly onto the glass, and then you put a barrier, a semi-permeable barrier, a barrier that is permeable to water and ions and small molecules, but the mesh is too tight to allow microglia through. So in this experiment, you have a physical separation of microglia and neurons, and you no longer get that degradation. So what does that tell you, chat? What does that tell you? Yeah, the insert is the barrier between the microglia and the neurons. Salmon, maybe? Yeah, thank you, Marlon. Yeah, I think it's salmon colored. That's an appropriate description. So when you add a barrier between microglia and neurons, you no longer see a degradation of alpha-synuclein within neurons, or a decrease in fluorescence. A sausage casing? Are sausage casings permeable to water? I don't think they are, right? They are pretty robust as far as I'm aware of. It's more like a net that has spaces that are too small for microglia to squeeze through, but small enough that water and anything that microglia might throw up can pass through. So what does this experiment mean, chat? What is the conclusion that we can draw here? So I described to you the data. Now I want a conclusion. What is our conclusion here? Seemingly not osmosis. Exactly, Marlon. So whatever effect microglia are having to remove the alpha-synuclein from the neurons is not mediated through osmosis or diffusion of molecules, right? Whatever is happening here requires direct contact between microglia and neurons. Do the membranes need to fuse? Yeah, so Marlon, you came in a bit late, so I'll show you this video. And forever who just dropped in, microglia, what they're doing here is tearing out the alpha-synuclein through these tiny little straws, so alpha-synuclein green. So you need this contact for this effect. And now, while we know that this can happen through direct contact, we haven't really proven that it actually means anything, right? So what this experiment tells us for certain is that this decrease, this 50% decrease in alpha-synuclein fluorescence, you need cell-cell contact for that to happen. And that is what makes this salmon bar such a good control, I should say. That would mean that the microglia are actively removing them through direct contact. Exactly. Very, very nice. Very, very nice set of experiments. So, while neurons can't degrade alpha-synuclein, right? These two bars, these gray bars, uptake and degradation, you give them alpha-synuclein, then you wait. They don't change their fluorescence at all of alpha-synuclein because neurons don't have the stuff to degrade them. While neurons don't have the stuff, microglia do, right? So when you have a burdened microglia, when you give microglia the alpha-synuclein, after 24 hours, it cuts that fluorescence in half and it just gets better over time, right? So while neurons can't degrade alpha-synuclein, microglia can. Through what mechanism? I don't know, but they can get rid of the alpha-synuclein. That's pretty amazing. So helping microglia help neurons might be a strategy to tackle neurodegeneration, right? Anybody who sees this working in biotech, I bet their brains are firing. I bet their synapses are firing right now. If we can get microglia to work better, maybe that's the way that we tackle these diseases like Parkinson's, like Alzheimer's, like Huntington's disease, what else am I missing? I think I got the big ones. So that's a very, very intriguing path for biotechnologists to go down. Side question, why don't neurons have the capacity to take apart alpha-synuclein? So alpha-synuclein is a very, very difficult compound to digest, right? And neurons are just not specialized for that kind of thing. Like they have proteasomes, they have ways to deal with burdens, but they're not professional digesters, whereas microglia are macrophages of the brain, and macrophages' whole function is to digest things like bacteria, right? They're hyper specialized to make the nastiest stuff the human body can make in order to break and destroy stuff. And so it's not surprising to me that microglia, which are specialized macrophages, have the stuff to really destroy this terribly difficult protein to break down. Is this mechanism always broken in those who suffer from degenerative neurological conditions involving accumulation of these proteins? It's a good question, Marlon. I don't know the answer to that. And these are just wild-type mouse neurons, right? So this isn't a disease model. These are just normal mice models. So if you give them alpha-synuclein, they're not going to be able to break it down. So that kind of implies that a normal, healthy person not prone to this kind of thing might be more susceptible if you artificially give them alpha-synuclein, right? Like, what might be preventing someone who doesn't get neurodegenerative diseases might be the initial initial aggregation, although I'm not entirely sure if that's completely correct. That's just where I would go with this line of thought. future biotech here seems like a very cool idea. Exactly. Like, in terms of my particular taste, I love basic science, so I stay in my lane here. But I do need to shout out everybody who wants to go into biotech, because they can then take these very exciting findings and then do something with it. And I think that's really, really exciting. Good luck, Exotic Fox. Very interested to see where your career goes from here. Do we know that those who have Alzheimer's have a higher level of alpha-synuclein? That would be a yes. So alpha-synuclein is a part of this mega jumble of aggregated protein that are in Alzheimer's brains. So whether or not disease is primarily driven by alpha-synuclein tau or amyloid beta, that's up for debate. But alpha-synuclein is definitely a part of the story. Great question, guys. So, microglia suck up toxic alpha-synuclein from neurons in a dish. We've already shown that. We've shown that it is meaningful. And now the question that we want to go towards is, how does this work? This is also interesting, but I'm already struggling at A-level biology. Here's the thing, Yan, I struggled with A-level biology too. Well, I mean, I'm not, I'm in America, so it wouldn't be A-level, but I think our equivalent is AP biology. In fact, I cowered out of taking AP biology, the exam, because my memory isn't so good. So just to keep it short, because I've told this story multiple times, yeah, my memory is really, really bad, but I love biology so much that I just kind of deal with it. And unfortunately, you have to deal with A-level and AP and intro level to be able to appreciate some of this cooler stuff. So let the cool stuff in biology be the passion and fuel that powers you through some of the more difficult aspects of learning this stuff for the first time. And I can sympathize, right? Like, A-level biology is a lot of memorizing information that you've encountered for the first time. But the more you specialize, the more times you get to see the information, the more it ends up sticking. So there is hope that it gets easier in the future, Yan. A-level bio gang, good to have you here. I'm always so happy to see bright students showing up in chat. It's very, very cool. Not my area of expertise. I'm more into ecology, but I love learning about that stuff. I love ecology too, from a very tangential perspective. We're all biologists at the end of the day. Isn't the strength of the immune system somewhat correlated to autoimmune disease? Wouldn't any way to increase microglia effectiveness run the risk of such a response in the brain? That's the thing about biology and biotechnologists, is that you just got to take the risk, right? You have to make a trade-off of... so yeah, there's no perfect medicine out there. Everything that you put in the body has a risk-benefit, and you have to make sure that the benefit of reducing neurodegenerative symptoms outweighs the risk of autoimmune disease. So while yes, that's not a reason why somebody should not explore this venue. All right. Brazilian, what do those levels mean? A-level, I think, is the equivalent of advanced studies in high school. So between the ages of 18 and, I forget, 15? Is high school. I don't really remember. But like, yeah, whatever that equivalent is for you. Thank you, Phi Sensei. You are very welcome. AP Bio Gang. Yeah, it's awesome that you guys are here. None of this will be on your exam probably, but I'm glad to see so many passionate young people. Okay, so how does this work? In order to understand how these processes work, we have to go down to the molecular level. Appropriate that we're talking about molecules here. And so I had mentioned earlier that the tunneling nanotubes are made out of a protein called actin. And so here we have a ribbon diagram of the actual actin monomer. So this is what it looks like at a molecular level. And then here we have a secondary structure of actin, or tertiary, whatever you want to call it. Those aren't the right words. Those aren't the right words. Here we have a superstructure of actin as a polymer, right? So each individual circle here is one of these blue molecules. And so actin kind of forms these strands, and these strands are capable of building up that cytoskeleton in all sorts of interesting ways. Here are two cells that have had their actin labeled. I don't know what cells they are, but you can see that this actin is present in the membrane and also present in membrane protrusions, right? So if you want to understand how actin works, or how tunneling nanotubes work, we have to understand how actin works here. Would you ever talk about your own research on a stream? I have, and I'll occasionally mention it, but in terms of doing a full thing on it. So the problem, Amina, is that my research was very neuroscience heavy. And in my opinion, neuroscience is not as accessible as molecular biology, because here's the thing about molecular biology. For how hard it is, at the end of the day, all molecular biology is molecules bumping into one another, and then those molecules do stuff, right? That's all biology is. And there's a lot of it, which is why it's hard, but the core of biology is quite simple. Neuroscience is a lot more abstract because you have to do math. And my research was in a field that it's got enough math ideas in it that makes it kind of hard to do for a YouTube audience. Although I've talked a lot about stuff that is tangential to what I researched. So membrane protrusion forms in the microglia and then enters the neuron cell membrane, exactly, and paths through the insides of a neuron. That might be going too far, Marlon. I don't know that we know beyond an initial connection what happens in these tunneling nanotubes. Well, there's also the thermodynamics involved, no? Yeah, I mean, when you're in school, you have to do a lot of that math to understand the basics behind it, but after you're finished with school, if you choose to do the science that I did, you only have to understand thermodynamics from a conceptual point of view and not a mathematical point of view, which maybe offers some hope if you're struggling with thermo. Of course, if you're an engineer, you still have to do math, but if you want to do basic science in molecular biology, you don't always have to do the thermo math, you just have to understand how thermo works. Okay, since tunneling nanotubes are made of actin, actin regulation is likely involved. So if we want to know the mechanisms behind how these microglia form these connections with these neurons, we have to look at how actin is regulated. And so here's one for the cell biologists, and here's, I don't really know, you A-level or AP Bio people can tell me if you ever go this deep into actin regulation. It's been very long since I was in AP Bio, but there are a lot of molecular mediators that regulate whether or not actin is in its filament form, F-actin, or if it's in its globular form, G-actin. And these molecules do things to each other. They do phosphorylation and other chemical reactions that send signals to either break down actin into G-actin or form these fibers. And these fibers are going to be very crucial for alpha-synuclein transfer. And so if we want to know how actin is involved, we have to start breaking some of these pathways to understand what molecular regulators are involved with this transfer. There's another biology VTuber I watch who once showed a picture of the metabolic map. He managed to scare half his viewers. Metabolism is crazy. I'm also very curious as to the other biology VTuber that you watch. I only know of like me and maybe 2 other people, so I'm very curious who you're talking about, Seedyum. No, we don't. That'll be fun for you in undergraduate then, or graduate school. I don't remember when you learned this. As far as I know, A-level doesn't go into actin at all. Oh, interesting, interesting. What's with the coloring? Yeah, so the blue is the pathway that this paper decides to investigate, the RAC-PAC LIMK cofilin pathway and the ARP23 pathway. I'm not going to get into what these do. That'll be for a Cell Biology 101 stream. But essentially, whether or not you get F-actin or G-actin is regulated by a whole bunch of cell biology stuff. And so this is one for the biotechnologists, because if you want to make this better, the interaction between microglia and neuron, you have to know how that interaction happens. So, in order to do that, we're going to poison these cells with inhibitors. I should have made this a question for chat. That would have been really interesting. Here's the thumbnail picture. If you have no treatment, you have a microglia and a neuron here, and it's making these tunneling nanotubes. And then we are going to poison RAC, and we are going to poison PAC with these molecules, EHOP and AZ. So if you go back into this figure, here's RAC and here's PAC. These are 2 components of the signaling pathway that controls whether or not actin is forming. And we give them poisons. And chat, I want you to practice describing to me what you are seeing in this data. What are you seeing in this data? When we poison this particular, because there are other components, right? There's CDC42, there's Rho-ROCK. When we pick a pathway and we poison that, what do we get here? What do we get? I hope that the TNT in the previous image doesn't stand for trinitrotoluene. It's not, and you can do exclamation point jargon to find out what it actually does mean. It's tunneling nanotube, these tiny, tiny connections that microglia form with neurons. EHOP is boba, AZ is Kiki. That's the second boba Kiki interaction I've had on the streams, which is very, very fun. He explodes. I'm seeing fewer TNTs. Yeah, for sure. I wouldn't say exploding is requiring more information than you have here. Describe to me the shapes that we're seeing here. The differences in shape, comparing no treatment to EHOP/rack poisoned to AZ/pack poisoned. More globular, that's a great way of describing it. No longer spiky, that's also a great way of putting it. EHOP spherical cell, no TNTs at all or protrusions. Yes, exactly. And so here's something that I always tell my students to do, and it's a non-trivial part of biology, is you gotta be able to look at data and figure out a way to use your words to describe it. And it's one of my favorite parts of the job. But it's often hard to get students to actually, because they just want to be like, what does the data mean? I don't know. And it's like, well, before you can understand what the data mean, you first have to actually tell me what is different about the data. And so, yeah, you don't see the tunneling nanotubes anymore. They're no longer spiky. It seems like rack is more, the EHOP poison is a little more dramatic than the AZ poison. Excellent observations that chat has made. Smoother, yeah, for sure. So that's not surprising, right? Given we know that these protrusions are comprised of actin. So if you break the actin pathway, either by breaking rack or pack, it makes sense that you no longer have these cytoplasmic or the membrane protrusions. And it looks like rack is more important than pack. That's not really surprising because rack is earlier in the story here. Great. There seems to be a lot of high aspect ratio structures in the AZ cell, which EHOP seems to entirely lack. Yeah. It seems like the pack looks more like the no treatment, I would say, in the fact that there are still these like membrane protrusions, whereas the rack inhibitor, this is just a ball. Very, very interesting. Welcome, Light Blue Fairy, welcome to the stream. We're talking about tunneling nanotubes, these tiny cellular protrusions that help microglia suck poisonous protein out of neurons in neurodegenerative diseases. So an inhibitor for RAC does stop tunneling nanotubes from forming and stops the microglia ability to absorb alpha-synuclein, right? So this is another layer of confirmation. If you poison the cell, if you get rid of its tunneling nanotubes by using EHOP, then you see in those microglia that you no longer get that alpha-synuclein fluorescence. They're not as able to take up that poison. And again, you see here that when you poison RAC with EHOP, that the number of tunneling nanotubes is decreased. So like chat said, the tunneling nanotubes, they're very present in the no treatment. You add that EHOP poison, and that's reflected in the data. You don't see that anymore. The length of the ones that do exist, however, seem to remain the same. I have no clue what any of these big brain terms mean, as my job is basically hammer go bonk. You can do exclamation point jargon. There's some stuff in there that can help you with understanding what we're talking about here. So here are the results so far, and this is maybe a good point to get reacquainted with our results. So alpha-synuclein, this is a protein that is aggregated or bunching up together in neurons that are damaged by Alzheimer's. This causes oxidative stress, it damages the mitochondria, and that can cause the neuron to die. Microglia make more of these thin straws called tunneling nanotubes, TNT, with neurons that have been treated with alpha-synuclein. These tunneling nanotubes contain alpha-synuclein, the culprit for a certain neurodegenerative disease, and microglia use these tiny straws to suck alpha-synuclein up from neurons in a dish. In terms of how these work, we know that these tunneling nanotubes rely on actin, and that actin signaling can affect whether or not these tubes form. But this is all well and good, and here is a question that was posed earlier in stream. Does this actually matter in a real, quote-unquote, real-life situation? Does this matter in a living, breathing neuronal network? And that, to me, is important. What's EHOP and RAC? Again, they're two different poisons that poison this aspect of the actin pathway. This is all cell biology, and I don't want to get too caught up into it, especially because cell biology is very hard. But it's also just, I wanted to give you a flavor for the biotech people out there of what doing biotech would involve. And it involves figuring out exactly how things work so that you can improve them in the future. Okay. So does this matter in an actual living, breathing neural network? So we have this experiment, and it's an interesting experiment, I'll say, where we take a mouse, a living, breathing mouse, and we open a window into its brain, and we engraft neurons that have been pre-treated with alpha-synuclein, and we check to see whether or not those microglia are doing what they do in a dish in real life. So engrafted neurons, we took exogenous or outside neurons, put them into a real mouse, and we checked the microglia afterwards. So before we get into interpreting this data, I want to ask chat, how good do you think this experiment is? Do you like this experiment? Do you have issues with this experiment? I want to think critically about the pros and cons of this particular approach. Again, taking neurons from the outside and putting them into a real life mouse and checking the microglia after that. So before we get into the data, let's think critically about if we like this experiment or not. Give you all a chance to think. Is there something concerning about this approach? Giving my teacherly nudge. Certainly an interesting point, certainly interesting question. I'll give y'all a little more time to respond because it is, I think, an interesting experiment to do. And for those of you who just want to know what's going on here, in red we have alpha-synuclein, in green we have CX3CR1, this is a marker for microglia, and in blue we have neurons. So when we're looking at green, we're seeing microglia, blue is neurons, and red is alpha-synuclein, which means that if you superimpose red with green, when you see yellow, that is alpha-synuclein in a microglia. Hairday says the neurons could be damaged when you put them in a mouse. Marlon says, have the neurons undergone the necessary developmental processes to match? That's also an interesting aspect, whether or not there is compatibility between these neurons. What if the mice recognize the outside neurons as non-self? Also an excellent question, Flying Taco. Alexandro says, I like the experiment, but some downsides. It's invasive, which can result in who knows what in the metabolism of the mouse brain. Yeah, exactly. Great input, chat. Great input. I don't love this particular experiment. Engrafting neurons involves immune activation, right? The very act of opening a window into the brain is going to activate neurons. Activate microglia, right? This is something that is done in other experiments. I didn't have time to bring up that data, but just doing surgery on the mouse is going to activate microglia. It's going to cause them to be more active, more immunologically relevant. And so there's a layer of artificialness here that I'm not a particular fan of. I would have actually preferred if genetic manipulation if possible, but maybe getting those mice may have been difficult or not feasible, right? In my perfect experiment, you get a mouse that is naturally pumping out more alpha-synuclein, and then you observe what the microglia are doing in their brains, instead of this system that could potentially be pretty artificial. However, you do end up seeing the tunneling nanotubes anyway. If you look down here, it's very difficult to image. You can tell that there's just a wisp of fluorescence here. These are pixels that are basically barely holding on, and you can see a smidgen of yellow that is helping these microglia take up the poisonous protein from these neurons. So I don't love the experiment, but for what they want to show, I think it's okay, right? That this can happen is better than not knowing if it does happen at all. So engrafted alpha-synuclein neurons form connections with native microglia. So can these native microglia actually help these alpha-synuclein neurons? So while the in vivo stuff isn't very compelling to me, like, okay, I don't love that there's this engrafting aspect of this immune paper, but okay. I'm a little sad, but I will move on. and think about different ways we can assess whether or not this matters in an actual living being. Are there any chemicals that destroy alpha-synuclein? I don't know any off the top of my head. So here is what the researchers do. So we're no longer talking about a living, breathing mouse, unfortunately, because it's really hard to do those experiments, where we're going back to a dish. And in order to get a better sense for whether or not the microglia are meaningfully helping out neurons, we are going to be checking whether or not the microglia can help alpha-synuclein burden neurons with their signaling, with their ability to talk to each other, right? Because neurons fire action potentials and actually communicate with electricity. And so while it's a bummer that we can't do more in the living, breathing mouse, this is also a pretty good way to check for health relevance. Can microglia repair neuron network activity from alpha-synuclein damage? And so here's a video from G et al. In 2015 that shows the concept of measuring neuron firing. And so what you can do is, when you have neurons in a dish, you can label a calcium channel called GCAMP-PF6 that fluoresces when it's bound to calcium. So every time a neuron pops off, it'll release a package of calcium that another neuron will take up. And when that neuron takes up that calcium, you get a ping of signal. In our case, the signal is going to be green. So we're going to watch this video a couple times to get a sense for the shape of the data that we're looking at. So again, green, when you see green, that is a calcium channel that received a calcium ion. So we'll take a look at that. Yep, so we're getting pings of green. Those are neurons that are getting calcium. So another neuron has fired and they're taking up that calcium. So you can see that we can actually use calcium fluorescence as a proxy for neuron firing. And we'll watch the video one more time. Again, every time you see a ping of green, that is a neuron getting a calcium injection, not injection, but taking up calcium. I do think these videos are pretty interesting. All right, does that make sense, chat? Ping of green, that's calcium, that's a neuron firing. So in order to quantify that, we can take these spike models. So we can isolate a portion of the visible field and measure the green fluorescence over time. So what you're seeing here are the spikes of green fluorescence over time. So in a controlled network, so these are neurons that have not had alpha-synuclein added to them, you get this pattern. So ping, 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 ping. That's the pattern by which these neurons are firing. Okay, now chat. What happens to neurons when they are burdened with alpha-synuclein? Let's see, just as a check for understanding if you've understood the shape of this data. What happens to neurons when they are burdened with alpha-synuclein? Remember, in the control, ping, 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 ping. What about these diseased neurons? Oh, that's an unhappy neuron. It's pretty dramatic, isn't it? It's pretty dramatic. It's quite striking, right? Alexandros is absolutely right. That is not a happy neuron. But again, yeah, try to describe what we're seeing. The vibe is that this neuron is very unhappy. But what is the actual data? Describe quantitatively what the data look like so that we can actually compare. Vast decrease in pings, exactly. Much less, exactly. The pathway is blocked and the signal isn't able to be translated. That is extrapolating a little too much, Exotic Fox. So when I ask for data interpretation, I want something like what Marlon and Robert are saying, just not including an interpretation, which that is, to avoid picking a hypothesis right off the gate, right? So we want to make sure to be disciplined in how we're seeing things. So I'm not saying, Exotic Fox, that you're wrong about pathways being blocked, but that's assuming too much for the signal here, right? So it is more accurate at this stage of our understanding to say there's a decrease in the frequency of spikes, right? So what we're seeing is fewer spikes, and so the jargon that we could say, the appropriate way to describe that would be to compare frequency. So ping, 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 when it's unburdened, happy, healthy neurons, you give them that Alzheimer's protein, ping, and then silence. Absolutely insane. Is the alignment of the pings horizontally meaningful? So the spike here just references how large that fluorescence data was. So the brighter that ping is, the more calcium was taken up. For the purposes of this paper, I don't think the amplitude of the ping means anything, right? Like how big that ping is. We're more concerned with how many pings there are, because to be honest, I don't really visually see a huge difference in the amplitudes to begin with. All right. Now, what about when you have alpha-synuclein burdened neurons, these neurons that kind of have Alzheimer's disease, and then you add wild-type microglia for 6 hours? Describe to me, don't make an interpretation, but describe to me what effect that has on neuron firing. What's the time scale here? Sorry, Alexandros, I blocked that part. It's you can see the scale bar here now that I've revealed it, that this is over 10 seconds. That, this bar means 10 seconds. That'd be the vertical sizing, wouldn't it? Oh, across, this is just time then, right? So this is just time. Across horizontal is time. Amplitude is the intensity of the light given off by the calcium channel. Horizontal is like how the alpha-synuclein lines line up with the gap in the control. Yeah, this is just going to be frequency over time. I don't really know. Oh, you mean like the fact that there's this big gap here and then there's this periodicity here? Like the regularity? I think that's a little abstract, and in a dish, you're always going to have funky periodicities because you're not in a real neuron network. Not as many pings as control, but more than alpha-synuclein. Fewer pings in control, but more than alpha-synuclein. Yes, exactly. Variable frequency in the pings. Yeah, it seems, but again, I wouldn't read too much into that because we are artificially in a dish and not in a real animal. So the pattern of pings I would not be concerned with, but the frequency is definitely increased. Maybe not as much as control, but when you add those microglia, you can see that there's functional recovery of neurons, right? Instead of being ping and then just quiet, you recover some of those pings, and that's really, really cool. And so if you quantify that data, here we have frequency in hertz, right? 1 over the events, or sorry, events over whatever over time. You can see that. Actually, that is bizarre, because this seems to imply that. Oh, because the error bars are enormous. I was really shocked for a second. So let's go over the quantification here. So between these two bars, we have 4 significant stars. So as you have correctly identified, alpha-synuclein much more quiet, not firing as frequently as the control. And then when you add the microglia, it seems that you recover that frequency, and then yeah, it seems like it's even better than in the control, but that's not really what we see in the example data, which is actually kind of shocking, that when you add the microglia, they actually potentially, yeah, this is the average, I believe, in a box and whisker plot, although I could be wrong. Yeah, that's actually pretty striking that they may be firing even more than their control neurons, which is not necessarily a good thing. Wow. I wonder why that might be. That's actually pretty crazy. I think there's an issue here, though. Do we have something missing from here, chat? Do we have something missing? Yeah, the thing is they had to pick representative curves, and they might just have not picked a curve that was representative of the aggregate data, which is kind of a shame. But there is something that I do take issue with here, and is that this is confusing and like doesn't line up with this, and there's also something missing here. What's missing? There's an experiment that's missing here, chat, and I want to know if you can suss out what experiment exactly is missing. Wild type plus control. Excellent, Alexandros. Exactly. We have regular neurons, we have alpha-synuclein burdened neurons, we have alpha-synuclein neurons burdened neurons with wild-type microglia. Where's wild-type plus control? Like, do microglia just help neurons fire to begin with? I know from when I was doing these experiments that when I added microglia to neurons, that those microglia would actually hurt the neurons when I did it with my hands using my specific protocol. So I'm wondering if microglia in general help neurons, and that maybe takes away some of the explanatory power here, which is kind of unfortunate. The control and alpha-synuclein are so close to one another in terms of mean in this graph. Yeah, which means that this is maybe not the most representative curve, which is a little, you know, a little sussy. Overall, I still think fantastic paper, but this discrepancy is a little surprising to me. Yeah, control plus microglia. Great job, guys. What's interesting here is that even though tau was not that exciting in terms of the earlier parts of this paper, it looks like microglia also help with tau burden, right? So here we have those pings in the control, tau really hurts. And then here we see, this looks a lot more like an increase in frequency, right? Tau plus microglia for 6 hours. You do see an increase compared to this curve here. But in the actual data, you don't actually see a statistically significant difference between these two groups. Overall, just kind of bizarre here. So the results so far, this is good review if you're just popping in. Alpha-synuclein, a protein that is implicated in neurodegenerative diseases, causes mitochondrial damage in neurons. And so microglia make connections with these neurons to help suck out that poisonous alpha-synuclein. And we know that this happens. We have video of it happening. You can look in the video VOD if you want to see those videos, which are really incredible. And it looks like the microglia sucking up alpha-synuclein and tau actually does recover neuronal activity in a dish. Or rather, it is more correct to say that the microglia introduced are correlated with improvement because we don't have that really important extra control, which is kind of actually annoying. Yeah, pretty frustrating, but I mean, I'm still pretty amazed that the microglia can help with this burden. And so wrapping up to the beginning of this story about donating mitochondria, remember that alpha-synuclein is causing mitochondrial collapse, releasing poisonous oxygen into the cell. And so are microglia also donating mitochondria to burden neurons? And so we can label the mitochondria with a thing called mitotracker, and we can actually see if the mitochondria that originate in mitochondria end up inside of a neuron. And here we see that, you know, you at 0 hours, you just add the mitochondria that have fluorescent. Sorry, you add the microglia that have fluorescent mitochondria. For 0 hours, the neurons are not fluorescent. And over time, you see an increase in neuronal fluorescence, meaning that these neurons are taking up fluorescent mitochondria from the microglia, which is pretty amazing. Here we have an actual picture and a 3D reconstruction. So in green, we have a microglia marker. And in here we have neurites. Actually, WGA might just be a membrane marker now that I think about it. It might not be microglia specific. And in red, we have mitotracker. And in this 3D reconstruction, we can actually see in these tunneling nanotubes, these straws, that microglia are, M words. Mitochondria are in these connections with these neurons. So really love this image here. It does look relatively quick, right? Like within an hour, you already see a good chunk of fluorescence in there. Although fluorescence importantly doesn't tell you how many, it's correlated with how many. So I wouldn't think, you can't be like, oh, the neurons got a ton of mitochondria in 1 hour. It just means that the amount of mitochondria they get are increasing. And again, in this experiment, if you physically separate the microglia from the neurons, you no longer get that transfer. Although there's a little bit of transfer there, right? There's a significant difference between 6 hours here and 6 hours insert. Although I don't know if they compared this group to 0 hours here. Just kind of interesting that there's any fluorescence in those neurons at all. I wonder if this could be a trailhead for a therapeutic strategy to recover certain diseases of dysregulated mitochondria. Exactly. That's where my biotech brain goes. If, there are tons of diseases that have dysregulated mitochondria, and if you can help cells that are helping with burden cells to pass off mitochondria, that could really be really important in terms of those diseases. So microglia donate more mitochondria when neurons are tau burdened. I don't think they have the equivalent alpha-synuclein experiment, which is kind of crazy here. So here, if you have control plus wild type, so this is just normal connections, right? So it looks like microglia donate their mitochondria to neurons even without the presence of disease, right? So 0 hours, you introduce the microglia and they're donating mitochondria as a normal part of their job. But when they detect tau burden, it looks like they donate even more. And that's also really interesting. So, I had no idea that microglia as a normal part of their job were donating mitochondria, at least in a dish. It's really, really interesting. Very, very cool. Yeah, Marlon, maybe you could engineer cells out of good mitochondria and then seed them through the patient's body to replace the damaged mitochondria. Exactly. Like, that would be an incredible strategy for treatment. So again, damaged mitochondria leak dangerous hydrogen peroxide and O2 minus superoxides into the cell. It's no good, which is why we care so much about these mitochondria. So incubation with microglia reduces the oxidative stress of neurons. So again, this is repeating these experiments to make sure that everything is working as we expected. Here we have control, regular neurons. They're pretty happy. You give them alpha-synuclein. We already know that they get stressed, that they get, ROS is reactive oxygen species. This is harmful oxygen within the cell. We see that when you give them this Alzheimer's protein, that they do get stressed. 1 hour after giving them the microglia, they remain pretty much the same, but after 6 hours, that oxidative stress is reduced. And again, that effect is abolished when you physically separate the mitochondria from the microglia. So we have a pretty amazing image, a pretty completely amazing image of how neurons and microglia are interfacing to potentially protect against neurodegenerative disease here. Discord invite still valid? I thought I had a permanent link in there. And if that's not working, let me go ahead and put a fresh link in the chat. That's really unfortunate. Yeah, give me 1 second to do that. And yeah, we continue to have these conversations in the Discord. As of late. Thank you, Toxo, for the invite. Okay. All right, so the results so far, ton of results. Alpha-synuclein treatment, poisonous Alzheimer's protein that aggregates, stresses out the mitochondria. In response, mitochondria make tunneling nanotubes, tiny microscopic straws to suck out alpha-synuclein and donate mitochondria. And that prevents the harmful oxygen from destroying the cell from within. Very, very cool. So would microglia donating mitochondria be a method of neuronal repair? That's what I'm seeing here, that is consistent with the evidence that they have shared so far. So microglia, conclusion, are capable of healing alpha-synuclein and tau burden neurons by removing the offending protein and donating healthy mitochondria. So I did this in the last paper with the aspartame because there were so many interesting nuances here. And I want to do it again here for a much better paper. And this gives me the opportunity to just lump praise before I take questions about the paper on what I think and how I feel about what I read. Because while people might be like, oh, facts don't care about your feelings, your understanding of the facts actually do care about your feelings. So it's important to acknowledge, in order to stay as unbiased as possible, that you do have feelings, right? If you've ever had therapy, it's not wise to only acknowledge your thinking brain and not your feeling brain too. So I want to give you my impression of this paper. There was a lot of work being done here, very thorough paper, and they're very difficult experiments to do because, and I know this firsthand from doing similar experiments in my own lab, tunneling nanotubes seem to be very hard to image. And again, like growing neurons that are healthy and microglia that are healthy enough to have these tiny protrusions and to be able to be talented enough to image these very, very tiny structures is very, very difficult. The mechanistic thread can be further explored. There's a bit of mechanism in here with that cell biology part with those actin cytoskeleton poisons, and that leaves a very tasty thread for biotech people to pull at to make treatments. There's actual clinical relevance here, right? Microglia may play a role in alleviating neurodegenerative diseases, not only by reducing the aggregated protein in those neurons, but also by donating their mitochondria. This is augmented by very powerful in vitro results that show an electrophysiological readout, although now I think that isn't so complete reading the paper once more, right? Ideally, we want one more control there, which might be in the supplement. I don't actually know if it's in the supplement, but I would have preferred for it to be in the main paper. The cons here is that the engrafted neuron experiment in the mouse, that to me wasn't super compelling, although I think it was fine to show that microglia can do it in the brain. It didn't really strike me as very physiological. I would have preferred a genetic model instead, but I am understanding, right? Like, I want chat to appreciate the kind of careful nature of my criticism, because right now in the science communication space, there are science communicators that rail against everything in science, and if you know who I'm talking about, you know. And those people go way too far, right? No paper is going to be a perfect paper because science is really hard to do at all, and you can see that the pros of this paper, in my opinion, far outweigh the very minor con of this like very small experiment that did its job, but maybe not in the best way. And it's important to acknowledge that there are flaws in research, but you should not judge science by its flaws, but by its successes, right? We have incredible success. And this is something that actually I've been talking to Sideray about like the meta understanding of science in the public sphere, is that while there are controversies, while there are papers that might not be perfect, ultimately science does much more good than it does harm. And I think it's important to acknowledge here. Okay. I can't wait to see if this is true in the context of an actual animal, right? A great paper makes you want to know what this lab is about to do. And maybe they already have the answer, but they want to split the paper into two for some reason. To be able to see this happen in a real animal would be absolutely incredible. So this is kind of a con, but also a pro, because I want to follow this lab and see what they are going to do next. Okay, so many questions, and I'm only going to focus on two, but I will pause here to take questions from chat. And I might not be able to answer them, given I'm not the authors of the paper, but it can be about anything regarding the paper, about neurodegenerative diseases in general. I will pause here before I want to get into a very fun brainstorming session where we think about tunneling nanotubes, these straws that connect these neurons to these microglia. Any questions from chat? Or comments from chat, right? Like, what did you think of the paper? How did you feel going through the data of the paper? Again, I'm very into acknowledging that scientists are living, breathing, thinking human beings, and I want to get a sense for your reaction, because if you ignore your reaction, then you end up thinking like you know everything because you're thinking with your quote-unquote objective brain, and I don't want to raise a generation of future scientists who are too cocky, right? Acknowledging your personal acknowledgments or your feelings is an important part of remaining objective, ironically enough. A brief skim of one of the supplemental appears to have no frequency graph with wild-type control. Maybe they forgot, or maybe they used reference instead. I doubt they forgot. I'm more surprised that reviewer 2 didn't ask them to do it. I don't really know. How selective is what these straws grab? Yeah, I don't know. Apparently people have known about tunneling nanotubes since the 90s, but this is the first time I've ever heard of them, which is really surprising. And yeah, like in terms of like how do they form, that's what I want to know too, Marlon. Like, what is the molecular determinant for whether or not a tunneling nanotube forms? You can continue to give me your reactions to the paper, but I do want to start you off thinking about engaging your scientific imagination. So these are tiny tubes that are ferrying mitochondria and poisonous protein in between cells. And I want to ask you, how do you think this is happening at a molecular level? Like, what is enabling the mitochondria to unidirectionally pull alpha-synuclein from a neuron? And here is where it's really exciting, because you don't have to have a biology background for this. If you're an engineer, if you like thinking about how to engineer solutions, you can think of this as an engineering problem. There's this straw that connects these two cells. How would you engineer machinery to transfer mitochondria and protein between these two states. I was annoyed at the frequency data. Why was that bar so large? Feeling irritated by the seemingly missing stuff. Also wondering if there's a link between the tunneling nanotubes and the pili. Yeah, I had pointed that out at the beginning. Those are valid concerns to have, right? But I don't want to, again, I don't want to raise, because this is a very easy trap, and I was in this trap when I was in graduate school, is that like, if I found one thing to be annoyed at, I was annoyed at the whole paper. In my opinion, the amazing results in those other sections of the paper outweigh some of the more annoying aspects of the paper. So I just want to make it, I mean, it's completely valid to feel that way. But as you read more, I think you will find yourselves being more forgiving, especially when you start doing your own science, right? Like when you are behind the wheel, you will find how hard it is to have every experiment be the absolutely most robust way you could do things. Although, yeah, I am personally a little annoyed at those omissions as well, but not annoyed enough to have a negative view on the paper overall. Walking protein, yeah, flying taco, that could be an aspect here. How does it regulate what travels through the structure? I really want to know that as well. Pressure differential, very interesting, or mechanistically pushing and pulling it. So how would it mechanistically pull and push? That's what I want to get into. What if the way mitochondria is pulled around is similar to the way chromosomes are pulled around during mitosis? Yeah, so maybe using some cytoskeletal elements, maybe being dragged by a thin thread. There's an excellent hypothesis here. Seemingly mitochondria are able to travel through the tube. Yeah, and I don't know why. So yeah, flying taco, that's actually, and Hair Day, you have come up with different mechanisms that I actually did not come up with. I want to give you all a little more time to think while I actually throw together an additional slide that captures flying taco's hypothesis, because it's actually not one that I thought of, and that's very exciting because and flying taco, and I think someone else had mentioned what they think is happening, and it's exciting because, you know, presumably I have more biology experience than you, but that doesn't mean that you can't have an idea that I haven't thought of already, which is very, very exciting. So what else? What other possible ways? And so, like, you know, why this is useful is because every hypothesis that you form can be tested, and that's important to recognize. The TNT that goes and just grabs it within the neurons, yeah, but like how at a molecular level is that happening? How does it localize the things to transfer through the tube? Yeah, that's also an interesting piece of the puzzle that I'm not aware of. This chat is going to make a breakthrough in the future. I sincerely believe it. You up-and-coming biologists might hold a lot of potential, especially because you're so young and so deeply engaged. You're much more engaged in biology than I was at your age, for sure. All right, so we have a couple of ideas in chat, and pressure is an interesting thing that I think I have kind of illustrated in my thoughts, but I want chat to think about the ways that I thought this could work. So here's my first idea. This is the idea of contractile rings. So we know for certain that tunneling nanotubes are made of actin. And this is kind of some background information. Actin can form these contractile rings, these rings that can contract and shrink, right? So maybe in this tube, sorry, it's not even on screen. Maybe in this tube, we have these rings of actin in red that can contract and expand. So maybe there's a mechanism, and I'm illustrating the alpha-synuclein protein here, where a series of contractile rings squeeze the protein in one direction. So maybe there are waves of squeezing rings, like when you brush your teeth, right? You squeeze at one end of the toothpaste, and then you go up the tube, and maybe there's a kind of toothpaste mechanism, right? Contractile rings. Any questions there? How that might work? So you have these tubes, and maybe there are rings in the tube. We know that they're made of actin, so it's very plausible that you can have these rings of actin squeeze in a wave of squeezing like a toothpaste tube to push the protein in a certain direction. Perhaps like peristalsis. Yeah, exactly, like peristalsis. So for those of you who don't know, peristalsis is an autonomic reaction that your digestive tract has, where it squeezes in waves to push food through your GI tract. That's an excellent metaphor for what is happening at the cellular level. And again, this is my hypothesis. So I like this hypothesis because we know that these rings can form in other contexts, so maybe this is what's happening here. I have another idea. Maybe it's actin motors. So some of you in chat have talked about walking proteins, and the protein that you're talking about here in the context of actin related stuff is myosin, right? So myosin is the motor protein that hinges on actin, and when it contracts, you get a squeeze of your muscle, right? So the way that that happens is you have these hinges, right? You can imagine like bumpers, right? Like if you're playing like a pinball or something, that these hinges can bend and sort of move the actin, move the protein in that kind of direction, right? So if contractile rings, or you're squeezing the tube in this direction, maybe these kind of bendy bumpers are moving back and forth and dragging that protein through, which would be really interesting. Yeah, so kinesin is actually specific for not the actin cytoskeleton, but the alpha beta tubulin cytoskeleton. So there's no reason why you would know that. There are a couple different systems of cellular support, one being actin and one being tubulin, microtubules, you might know them as. So contractile rings, I really had a lot of fun imagining what could be happening. So the toothpaste method, the pinball method, we could say, and then I have one more strategy that I think is kind of fun to think about. Maybe it's cytoplasmic flow. Maybe there's a, and we know that this happens in development for certain species, where you have the actual juice of the cell, the inner cytoplasm, that is being streamed into the other cell. So it's like literally like a straw, like sucking up the juice of the neurons. Instead of this protein base, maybe it's cytoplasmic, and it's just the liquid is pushing the synuclein through. I don't really know. I don't know what the answer is here. But these are all ways that actin could be involved, right? Either actin is in contractile rings, maybe actin is here, and the motor protein myosin bound to actin is pinballing the protein through. Or maybe actin is just the tube, and there's some other motor, maybe kinesin, maybe dynein, maybe myosin itself. They're moving the cytoplasm through and sucking the protein up the tube. All very interesting. Yeah, this would be like that pressure hypothesis. Exactly, like something like a pressure differential or moving liquids would be very interesting. What about Louise's suggestion? Let me scroll back to that. You bait the mitochondria, they're live symbionts. I don't know enough about how mitochondria function to be able to comment on that. I'm more focused on alpha-synuclein because I know more about protein than I do about mitochondria. Yeah. So here we actually have fish tacos hypothesis. And this is like if there's direct binding between an actin filament and alpha-synuclein. Maybe there's something at the end of the cell that is dragging the alpha-synuclein, like a fishing rod. We'll call it the fishing rod hypothesis of pulling the alpha-synuclein through. I genuinely don't know which of these versions are happening. And we don't, I think, in this paper have enough information to be able to tell which of these are happening. And now that you've seen my hypotheses and Chad's hypotheses, I want to end by just kind of brainstorming how else this could be happening. Like, I don't think this is, I don't think this is extensive, right? I don't think I've listed every single engineering possibility here, but what I will say, just in order to like do additional brainstorming for a similar like next direction thing here, is that how would you test these hypotheses, do you think? How would you test these hypotheses? And this is more of a question for my undergraduates in biology rather than my A-level or AP biologists, because I'm not expecting a non-biology person to be able to tell me how to test these potential models. And we'll go with the three that I had listed here just for simplicity's sake. But some of my biology people, I don't know if any of you are in chat, but how might you, or anybody can participate here. There's one of these, or rather there are a couple of these that would be testable in terms of mechanism. tags, lots of fluorescent tags. So what would you tag? I want to encourage you to think a little more deeply about what you would tag here. And there's trouble here, right? Like we've already seen that fluorescently tagging tunneling nanotubes is really hard because they are so small, you can barely see them with fluorescence. Maybe the tubule is building items on one end and disassembling items at the other end. Yeah. So maybe you have kind of a treadmilling action going on that's pulling a filament in some direction. But how would you test whether or not contractile rings or actin transport or flow is responsible here? And I'll give you a hint. This paper took a similar approach when they wanted to test the role of actin in this system, tagging the actin monomers. Again, tagging isn't going to really work in this case because the resolution of fluorescence is not good enough to get extreme detail here. Or rather, tagging actin might not be very useful, but maybe tagging something else could be better. In the flow, the cytoplasm in the rings tag the mitochondria and analyzing how they move. So if you were to tag them, they would all just look like what we've already seen in this paper. They're just tubes. You can't actually get that high resolution if you are just labeling actin, right? Let me see if I can actually illustrate this by going back further into the paper. I want to really emphasize that these are very hard because these are already, not this one. We already have what it would look like to label actin is what I'm saying. So you wouldn't really get additional information. I think red here is actin. So if you were to label actin, this is what you would see, right? We actually can't, I don't know if you could come up with a way to tell if this was contractile rings or a bumper or a fishing rod or anything, but they're just little thin lines of red wisps. Because they are very challenging to image. So fluorescing is maybe not the strategy I would personally go with, but I might be wrong, right? There could be a more clever way to do it that I'm not really aware of. Can we monitor their electrochemical signals? Which ones? What might that look like? And how would that narrow down the possibilities? So I'm here discouraging labeling actin, but what else could we label instead of actin here? Actin makes the tubes and the motors. So we're getting there. Actin attaches the motors to the protein, right? So maybe that wasn't clear, but actin is the cytoskeleton and myosin is the motor protein. So what does that tell you? What could we label instead of actin? What could we label instead of actin, do you think? Also, for the contractile rings, in order for these rings to contract, they need myosin as well, right? Because it's the action by myosin that allows actin to change structure and to shrink like this. The protein, yeah. Tagging myosin, exactly. We could potentially tag myosin. Again, these structures are so small that even tagging myosin might not be the smartest idea because those tunneling nanotubes are super, super thin. So if we can't tag myosin because the location is so small, what else could we do to myosin to test the hypothesis that you need myosin to move this toxic protein? What could you do to myosin instead of tagging it? Tagging it would be nice, but there's a practical reality here, and that's the tunneling nanotubes are too small to image reliably, especially not in a low abundance protein like myosin potentially could be. This is challenging, and I think you're going to be really proud of yourselves if you can figure this out. Less myosin, Alexandros is on the right track. Myosin knockout, Marlon, that's a very good idea. I will go in between Alexandros and Marlin because if you knock out myosin, there's a good chance the cell doesn't survive. Inhibit myosin, yes, Exotic Fox. This is an approach that this paper took earlier with the actin regulation system. By poisoning specific compartments, you can have a targeted understanding of how things work. So you could just do this for all of the motor proteins if you really wanted to, right? Say, for example, you put down neurons in a dish, you added microglia, and then you added a myosin specific poison, do you still get transfer of alpha-synuclein, yes or no? Let's say you add a microtubule, right? Because cytoplasmic flow, while these two hypotheses here are driven by actin-myosin, cytoplasmic flow could be from other motor proteins like kinesin or dynein. So you could systemically test each motor protein poison to see what is required, what is necessary for the transfer of protein through these tubes. Good job, y'all. Very, very smart. I hope you are proud of yourselves for coming up with that. With a bit of guidance from me, but just a little bit of guidance, you have come up with what I would think would be the next direction to go to, because I would love to know what the heck is happening in these tiny, tiny tubes, right? What is happening? Sorry, I have to go so far back. What is happening? You can see here, it's very hard to image, so labeling is not a good idea. But what is actually happening here in these tiny tubes that allows you to move alpha-synuclein from cell to cell? And I would be really excited to know, and I would be really happy if if any of these hypotheses were proven true, right? Because this came from my own brain, and then this came from the brain of chat. And if chat gets it right and I get it wrong, that would be amazing. I would love to celebrate that, a member of chat coming up with a smarter idea than what I came up with. All right, that is the end of this paper. Any additional questions about the paper, or I'll just open up in general the questions in general, because I missed you all. I didn't get to stream last week, so I want to spend a couple of minutes just chatting with all of you, checking in on how you have all been doing, and how, if you're in school, how the semesters have been going. I'm very, very curious. Alpha-synuclein is moving from the neuron to microglia, and mitochondria are moving from microglia to neuron. Exactly, and that's really interesting too. I don't know enough about mitochondria to know how they could be experiencing unidirectional transport, right? There's the entire mitochondria story that I kind of ignored, and that Louise rightfully wanted to draw attention to, but I kind of ignored because I was running out of time, honestly. But yeah. I mean, the fact that one particular item is unidirectional could be due to the intrinsic structure of alpha-synuclein. Maybe it only attaches to a certain part of myosin, and that part of myosin is directionally specific. And then the mitochondria, I don't know if there's like a bait that mitochondria react to. I just don't know. That would be really interesting to know whether or not there's some signal in the neuron that asks the mitochondria to swim over, like chemotaxis, for example. Chemotaxis being the process by which cells can sniff out chemical signals and move towards them. Something that neutrophils are very famous for doing, by the way, reacting to chemicals in the environment and heading towards that direction. I have a question. I have a terrible fever, sore throat, and cough. Is it viral or bacterial? Not a doctor. I would not be able to answer that for you. Unfortunately, I am a biologist, which means I can tell you all about the bacteria and virus, assuming I know of them, but I would not be able to give you any kind of health advice or diagnosis. I'm being owned in my undergraduate course. On the plus side, haven't failed anything so far. Undergraduate is tough, man. The volume of information that you have to intake as an undergraduate, I have a lot of respect for. It's like a fire hose sometimes, but I believe in you. If you are the type of person to show up to the Find the Neutrophil stream, I believe you have what it takes to get through it, I should say. And on the other side, it's a lot nicer too, because once you don't have to study for tests anymore, you can just study on your own, like I do for this channel, and it's a lot of it's a lot nicer that way. I feel pretty bad. I'm so sorry. It sucks to be sick. I've been wearing my mask in public, very, very frequently, because the current strains of the flu and RSV are very, very tough on the human body. And if you have any one of those, I really feel for you, because I've heard reports of the current flu going around in America being just a nightmare to deal with. I had a frustrating lab mate yesterday when trying to do a Bradford assay. Oh, what was frustrating about your lab mate? So for those of you who don't know, the Bradford assay is a colorimetric, meaning it produces a color, assay to measure the level of protein in a particular sample. So I believe Bradford is a measure of blue absorbance, and so the more blue your sample ends up being, the more protein you end up having. A-level mocks in 2 weeks. Good luck, Flying Taco. I'm sure you will nail it. Although, again, better person than I because I just didn't end up taking my AP exam. Haha. But again, it shows that like you can still be doing biology and kind of suck at A levels and APs as I did, because again, it's just a fire hose of information, and not every teacher does a great job at explaining biology. My philosophy for teaching is that biology should be taught as a series of stories, because stories is how humans learn, right? You don't learn by looking at slides and just brute force memorizing everything, but you learn by compartmentalizing things into stories. And the more you can do that, the better. And I feel like I did not have that experience in AP biology, which is why I did not have the confidence to actually take the exam, nor the guts and willpower to study on my own. Yeah, so certainly good luck to all of you for your exams. Yeah, the students that I've been teaching have been under midterm stress as well. You can actually see it on their faces. I feel so bad for them. That they are good students, but when they are midterm stressed, you can kind of pick up that they're fighting for their lives out there, and I do feel for them. So good luck to all of you who are in midterm season right now. All right. With that being said, I am all ready and set to go. Because I am very hungry and I would like to get some lunch. There will be some news for the channel that will be dropped either in the next week or the week after. And I've got all sorts of emotions and feelings about what that news is going to be. And I just wanted to cue you in on it. It's nothing that is it's certainly interesting news for me in my personal existence that I will disclose next week when I figure out exactly how I want to communicate that information. But be on the lookout for a big announcement sometime in the next 2 weeks. Yeah. And I'll take a couple last questions and then I will head off to lunch. Love your contact. Thank you very much. A second year uni has some interesting modules that I'm looking forward to: microbio, ethnobotany, and food technology. Very, very exciting. Microbio seems very, very exciting. I would love to take a microbiology class myself one day. I have a question though. Do you think immunology is a good biotech direction, or would you have to go through something like AMR research? I don't know what AMR research is, but immunology is a fantastic biotech direction. Immunology is so hot right now in terms of like leveraging the immune system to fight all sorts of different things like cancer and infectious disease. Is so incredibly important and innovative right now. And if you can find an institution that is still investing in that, I would go nuts. It's so good to be an immunology researcher right now in terms of developing treatments for all sorts of terrible diseases in humanity. And with that note, I wish you all good luck in your studies and your careers, and I will see you at some point in the future. Good luck to all of you in all that you do. Goodbye, and thank you for watching. Bye, y'all.